Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. In this episode, I talk to Dr. Antonella Di Pizio from the Leibniz Institute for Food Systems Biology at the Technical University of Munich in Germany. She is a computational pharmacologist who studies chemosensory receptors. Her postdoctoral training in Dr. Masha Niv's lab in Israel led her to first working on GPCRs. She focused on bitter taste receptors. Now that Antonella has her own group, she uses various computational tools to better understand taste and odorant receptors, as well as trace amine receptors. Antonella is our very first computational pharmacologist guest on the show. If you are or know a computational scientist working on GPCRs, we'd love to chat with you on the podcast. Reach out to us at drgpcr.com/podcast. And now, let's dive into our episode with Dr. Antonella Di Pizio. Hello, listeners. I'm Dr. Yamina Bershish, founder of Dr. GPCR. And on today's Dr. GPCR podcast, we have the honor of having Dr. Antonella Di Pizio with us. She is an independent research group leader in computational pharmacology at the Technical Institute of Munich, more specifically at the Leibniz Institute for Food Systems Biology. Hi, Antonella.
Hi, thank you, Yamina. My pleasure.
I'm so happy to have you with me today, especially with the whole time difference.
Yes, but we manage.
As I just mentioned, you're a computational pharmacologist. Can you tell us a little bit more about what you do and how you got to where you are today?
Sure. As you said, my institute with this long name, so it's an institute that is focused on food system biology. And it's affiliated with the Technical University of Munich. And it was founded 1 year ago as a food chemistry institute. But then things change, and they discovered that also food is more complex. And so they wanted to have a computational section. So last year, actually, I joined in October 2018. My lab was the first group in the in-silico department. And the computational pharmacology is somehow this name is trying to define my group as looking at the interface between the food and the pharmacology that this food can have, these food ingredients, but also because I look at the receptor level of the compounds. And specifically, the Institute is very focused on the reception of food, so taste and smell, and I look at the smell and taste receptors.
That's so interesting. We oftentimes forget that, to taste and smell, we also use GPCRs, obviously. They're everywhere. So how did you get into the field of GPCRs?
Yes, by training, I'm a medicinal chemist. So I obtained my master in Italy at the University of Chieti. And then I started my PhD in computational medicinal chemistry in the group of Mariangela Gamennone. And the focus on my project inhibitors of matrix metalloproteinases. So I was working on different targets, but there I started my training as a molecular modeler. So to use all the computational tools from structure-based, ligand-based, so docking, pharmacophore, molecular dynamics, any kind of tool that you can use to design new compounds. And then I decided to go for an European track of the PhD. So this means that you have a European committee, but mainly that you have an additional supervisor and you spend a period in a different lab. And in this case, we decided to ask to Professor Gerhard Klebe, who is a medicinal chemist in Marburg in Germany, and he strongly advised me to do structural biology. So I put my hands in the lab and I got my own proteins. And this was an amazing experience to get familiar with the structures that I usually was used to look at the computer. And then, all this background, all these competencies just finalize my PhD thesis, and then what to do next. So I tried to answer this question, and the answer was in Israel. So I ended up in Israel in the lab of Masha Niv. So she works on taste GPCRs, and this was my first meeting with GPCRs, starting from the bitter side, because I started to work with bitter taste receptors.
So interesting. And I have to tell our audience that Masha will be one of our future guests on the podcast, and we're excited to have her as well.
I mean, this is a kind of mentor-mentee relationship that is continuously. So I'm always in touch with her, and I knew that I will anticipate on some of our previous results together today.
Right now, you look at the computational pharmacology of GPCRs, of taste receptors, and you're interested in bitter receptors. Am I correct?
I mean, this was my postdoc. It's still current interest, but now I'm trying to enlarge also to the odorant receptors. So I say I try because the odorant receptors are really complex. So it's the biggest part of the GPCRs. So I have 400, but also I'm working in a smaller, a more simple repertoire that are the trace amine-associated receptors. And these are just 6. They are expressed in the olfactory system, and they respond to amines. But as the name says, they respond to trace amines. So they are in the interface between smell perception, but also neuro perception, because these trace amines are neurotransmitters in our body. So we have, for example, the TAR1 that is expressed only in the brain. So we don't find anyone in the odorant system, but we have the other 5 that are expressed in the odorant system. And then we have some that are both in the brain and the odorant system. So, this were the perfect match of my interest in both drug discovery, but also in food and in general in the recognition of molecules.
Yeah, and what's the use of having these receptors expressed in the olfactory system and in the brain? You said there were 6 of them. First of all, what's the use? Second of all, what are the commonalities between all these 6 receptors?
One of the reasons why I get interested in these receptors is actually more structurally, because these are class A GPCRs and they share a similarity with known crystal structure, the one that are already determined, that is around the 30%. So, during my postdoc, I worked in these bitter taste receptors that share from 5 to 10% of identity. And this was a huge challenge. So when I saw 30%, I said, okay, this is another step. So this is something that it's already a good similarity to do computational work. And these are somehow similar to other GPCRs. So this number that I mentioned, like the 30%, is with the beta-2 adrenergic receptor. So structurally are more classical GPCRs. And functionally, there is not a lot known, but a lot of interest. So the TAR1 in 2019 was defined by the FDA like a breakthrough treatment for schizophrenia. So because there is this new molecule that activates this receptor that is like a breakthrough in the area. So these are the TAR1 that is the most studied pharmacologically is thought to be a relevant target in neurodegenerative disease and neurological disease. For the others, there is a lot to do, and the main thing the basic thing that we need to do is to find more ligands. So this is why I think that computational chemists can help, because, for example, the TAR5, the one we are currently working now, we have just very few agonists in the number of 5. It's not 500, what we are, the number that we are usually thinking in a pharmaceutical order. So it's really almost an orphan receptor. But still, when it was recently found that when it's expressed in the brain, it alters the serotonin levels. And in mice, when you have a knockout, you see this behavior like anti-anxiety, antidepressant behavior. So an antagonist of this receptor can be used, in future can be developed as for depression, anxiety, and all these psychological disorders.
Oh, wow. Fascinating. Yeah, so there's five ligands. You can count them literally on one hand in this case. If you had to choose one favorite receptor or one family of receptors, with your eyes closed, saying which one would it be?
I have many to choose, so no? So I will, maybe it depends on the time. I mean, because as you were saying, GPCR are everywhere. So if I wake up, for sure, I would like, my favorite receptor is one of the odorant receptor that make me smell the coffee. But I also like a lot bitterness of the coffee. And so it's just, I really like this chemoreception in general, because you have these GPCRs working together for your flavor and your taste. And this is, I think, just something that we experience, and so we know that taste and that flavor is a complexity of different signals, but I think that it is the same in our body, what we don't see. So unraveling this role of the communication of the different receptors can somehow be the key also to understand more the GPCR world in general.
It's so interesting with the taste. I was talking to someone and they were mentioning that some people have mutated taste receptors that make it so that they don't like cilantro, for example. I think to them it tastes like dirt, which is very interesting. About it, it's unique, but it's very, very interesting.
I just think that this is a field that is sometimes underestimated, because of course diseases are sometimes, are more important. Is life or living or dying. So it's something that always is considered less important. But we discovered also with this pandemic that actually we had this loss on taste and smell, like one of the primary symptoms of the disease. And it's really connected, the biology is we are one body, and there is a connection of many different receptor molecules in our body. So studying this receptor is important also for diagnostics. So there are many connections on taste and smell with disease currently.
Definitely, I did see on PubMed your paper about the taste receptors and the current pandemics, and it being one of the symptoms. What can you tell us about what's known on taste receptors in the context of loss of taste in the COVID pandemic?
So there is a partial knowledge until now. Everything is partial knowledge about this COVID-19 until now. And so what we know is that also in the olfactory epithelium, we have the expression of the S2, so the receptor of the virus. But this is, so it's not connected with the GPCR, with the odorant receptor, but it's like that the virus, when binds to the receptor that is present in subcellular cells, then you don't have, you have like a stop of the smell at all. So you can have a complete anosmia. So it's a cellular damage. And it's not receptor based in the sense of the odorant receptor, because the neuron is not affected. So the idea of the last paper was to have a perspective review of what is known, because actually this is not the first virus that affect taste and smell. It's just that this is maybe the first virus that affect the world together. So we have huge numbers, but the idea is to understand if this virus is different at a molecular level, or if it is something that is more related to a common viral infection, because virus can damage the olfactory system. And in this case, we have a lot of evidence also for the taste system.
Wow. And you had mentioned that other viruses also can damage the olfactory system and the taste system. What are these viruses?
Many different viruses is not something. So I'm currently, so when the pandemic started, we just organized this consortium for studying this phenomenon, and I just volunteered for the Italian team just to organize, a questionnaire for a language thing. So I gave my email to help and to collect data and to spread the questionnaire. I'm receiving so many mail of people that just lost their taste and smell suddenly, just with the normal flu, and they just couldn't get it back because sometimes it's something just transient, and sometimes you lost, without having it back, never. And it's really serious. It's not something you say, okay, I don't smell my coffee, and that's okay, because it's really affect your lifestyle.
Yeah, it does. I mean, eating and having pleasure and being able to taste all those foods is so important psychologically as well. And I cannot imagine not being able to smell or taste the coffee in the morning.
Yes, what I was mentioning before, coffee is so important.
It is. It is. I think a lot of our audience, a lot of scientists can understand the importance of having the right amount of the right quantity of coffee to be able to function during the day. So we're talking about taste receptors. What is the current knowledge base on taste receptors in general? 'Cause you had mentioned that it's important. Not everyone has been working on it, or we don't have enough evidence or enough information on these receptors. But what's the status of research today?
I look at my perspective, So, I mean, bitter taste receptor, if we want to talk about bitter taste, are of course important mainly for food. So, for years, the idea was to find something that to make food more pleasant, the interest of food industries. So most of the research focused primarily on the chemistry of these compounds. So to identify these compounds, to try to block these compounds, and the receptors were identified just in the 20s, the 2000. So from this year on, we started to have a more receptor-based research, but we don't have any crystal structures. So the research is more food-oriented and food chemistry-oriented, and this is why we have knowledge about compounds, but we don't have a lot of knowledge about, for example, quantitative structure-activity relation studies based on the structure of the receptor. So currently, for example, the bitter test, we were saying that odorant receptors are many, and I mean, this is pretty well known because there are 400, but also bitter test receptors are 25, so we don't have one receptor and many compounds. So we have a repertoire of receptors that are very different among them. So the sequence identity range from 15 to 19 among the same family. So we have very couple of bitter test receptors that are very different among them. And this is a lot of material for a structural biologist, and for a structural bioinformatics, so to analyze the sequences, to analyze the 3D structure. But until now, no one invested in the structural biology of these receptors. And this is also, and also no one invested in finding the most potent compounds to help also the crystallization. And this also makes sense why you should want to have the super bitter compound. So it's something that somehow doesn't go in the same path of other GPCR that we may know. So we have more fractionated informations. What we actually did when I was doing my postdoc is trying to fill some of this gap. So to try to use virtual screening to find compounds that can accommodate the receptor in the best way, and this in order to help to characterize the binding site.
So on a day-to-day basis, you work in front of a computer, you have a set of structures, and what do you do? Can you tell us a little bit more in practice? Let's say you're interested in a taste receptor. How do you use the current information available in order to get more knowledge on bitter taste receptors, for example?
Yes, there are many different levels, because here we have a different level of complexity. So one can be receptor based. I don't know, you have one specific receptor, as I was mentioning before, and we want to understand the binding site of this specific receptor. So in our case, what we were working is the TAS2R14, because this receptor can be activated by almost 50% of the compounds that are known to bind bitter taste receptors. So this means that it's an important receptor for bitterness. But in other case, you might be interested in understanding the molecular recognition in a higher level. So you want to understand why this receptor can be activated by so many compounds, and instead you have another receptor that can be activated just selectively by one or two compounds. In this case, you will look at all the repertoire of the receptors. And this is also something that we did just using different tools like bioinformatics tools applied just to machine learning or linear regression, some statistical method that help you to correlate properties with function. And in this case, the function will be this ability of the receptor to bind many or few ligands. Or you may want to find antagonists for a specific receptor. But in all these cases, the approach that I basically and routinely I use are the basic tool of the computational chemistry that are used in pharma. So you just use homology modeling, docking, and then you can use ligand-based tools. You can calculate all the molecular properties of the small molecules, ranging from the physiochemical properties to topological properties. And you use these nice tools that are routinely used in drug discovery just for a different purpose, that in this case will be to understand the activity of bitterness.
And then once you find suitable compounds, for example, you've looked at a receptor that has 50 different ligands, and you try to figure out which one would be the best to induce this bitter taste. Do you translate this research into a wet lab setting, or are you collaborating?
Of course.
Okay.
Of course. So we have many collaborators, and in this special case, it's very important not only to go back to the experimentalists, but also to have iterative working with them, because from the beginning, so from the modeling of this receptor, since you have a very low sequence identity, you don't want to do all the computational work and then just at the end you will discover that your model was wrong. I mean, any model is wrong, but it was completely not predictive. So you want also to use experimental information from the beginning. So after the modeling, there are position in the binding site that we predict that are important, that undergo to mutagenesis, to experimental mutagenesis. And then you take back the experimental results and incorporate in your modeling. So I mean, it's a very collaborative work. It's not just computational, and then we go to the collaborator. And this is what I like it a lot, to have such a multidisciplinary approach in the project building, project management, and not only at the end of the workflow.
Yeah, and I think it provides, having the computational side matched up with the wet lab side, from different areas. I think it does help advance the research, because more brains mean more brain power, and more toolkits mean better understanding of the research that you're doing. I think it's fantastic.
And you share your research goal with your collaborators, so they are not you're more partners than collaborators. Yeah.
So we were talking in the beginning of the conversation about, I think it was an olfactory receptor, that was when it's knocked out in mice, you have less depression. And can you tell us more about that? I think it's a very neat model, and it's a very good disease target.
Yes, I mean, I mentioned this case because, I mean, GPCRs are so many and are so important that sometimes, we just look at these three, but then in our mind, we just keep thinking about these main players in the GPCRs, and instead, even these kind of orphan receptors that we are just discovering can be maybe the new target for important disease where we are still looking for drugs and where we still need for drugs. So here we are really at the beginning, It's just target identification, that this protein is doing something. And then we know, we likely now will have more structure, and we currently have many crystal structure. Cryo-EM is producing a lot of structure. So we have templates for our homology modeling, and each day we have more. So we really have good chance to use also molecular modeling to drug design, exploring a new area and new GPCRs.
Yeah, absolutely. Last month was a structural month. There were so many cryo-EM and crystal structures that came out. I think I counted 10 papers in a month, which it's just amazing to see all these structures come out. And for taste receptors and olfactory receptors, are there any structures available?
No, but any structure, is a piece of information in this puzzle. Because of course, I hope to see one of them very soon, because of course, to have a structure of this family will help. But even having a range of structures from other families can increase the description of this family. The different shapes of the extracellular loop, or the different architecture somehow, or even tiny details, and that you have just when you have many structure can provide inputs also in research in other different areas.
I like the fact that you said it's a puzzle. I also see it as this huge puzzle, and getting more structures is like getting more snapshots of inactive and active states of these receptors, and just putting it together will be useful to better understand the structures or the function of GPCRs, for which right now currently we don't have a structure for. But I really like it. It's a huge, very complicated yet very interesting puzzle. Speaking of puzzles, what other tools other than crystal structures you feel like we need more in order to better understand taste receptors, for example, and olfactory receptors?
Molecules, compounds as tools. All for the majority of these receptors, the researchers started looking at this receptor as the chemical senses receptors, as receptor for bitter taste, as receptor for odorant molecules. But actually, now we know that these receptors are expressed everywhere. So as I was mentioning, this particular example is expressed in the brain and in the nose. And the molecules that go in contact with the nose maybe are not the same one that are in the brain. And this is true for any of these receptors. These are expressed ecnomotopically, so out of the usual place. So we have odorant receptors in the intestine, in the brain, and then we have bitter taste receptor in the heart. So are really everywhere. And what these receptors are doing there, we don't know. In most cases, we don't know. Of course, there is a lot of research on it, because this is now an important question to answer. But also, which is the chemical space of molecules that this receptor can recognize. Now we know that it's still partial. So we have this puzzle now, because the odorant molecules that we can recognize with our nose may have different properties, can cover a different chemical space of the molecules that bind the odorant receptor in the intestine. And so we may have molecules that are metabolites that are actually endogenous ligands, and they may have different structure. So the more we know also about the chemical of this receptor, more information we will have also about the function. But in between, from the structure to the chemistry, there is the biology, how this molecule binds this receptor. We will know more about the molecular recognition.
Wow, that's fascinating. I didn't know that there were receptors in the intestines and in the heart. You'd think, why are the especially in the heart. What do you do with the taste receptor or an olfactory receptor in the heart?
The other question, are they only bitter receptors? So and it's many of the compounds that were found are like secretion of bacteria. So there is a lot of evidence that, for example, bitter taste receptor can play a role in the immune system, like an additional immunity part. So you can recognize molecules that may be taste bitter, but I mean that are actually secretion from bacteria.
That is awesome. And we could also hypothesize that these receptors could form dimers with other receptors, for example, in the brain. I think it's a understudied world, but it holds a lot of secrets that are definitely worth studying. So one of my questions in the list I had sent you was, do you think GPCRs are still good drug targets? I think we all agree that GPCRs are still good drug targets. But I wanted to hear what were your thoughts about what we know currently, for example, on taste and olfactory receptors and disease, or at least as drug targets. Does anyone target them?
Yes, I mean, what we were saying until now, I think that is already quite convincing, that I do believe that GPCR are maybe the drug targets. I mean, they are in the cell, on the surface of the cell, that they have this nice defined binding site. So computationally, they are the perfect drug targets. But also we discussed that there are many unexplored area and many potential of drugability that are then there is still a need to know more about these proteins, and there are many GPCRs that we still don't know, that I definitely think that they are drug targets. And just to answer more specifically your question about the taste receptor, so we have currently on clinical trial on phase 2 a class of compounds that are isohumulons. So these humulons are usually hop-derived compounds that are important for the taste of beer. So they give this typical flavor of beer, typical taste of beer that we really like. So it's a bitter taste that actually is not disliked at all. And, as many natural compounds, these compounds were found to be very good, and to be bioactive compounds, to be good in to have many properties. And one of the important properties was these anti-diabetic properties that they had. And of course, you have a natural molecule with an interesting properties, and then there is some therapeutical indications. So drug industry can get interested. This is not something new, it is a flow that we can tell many stories about it. But what was interesting that after this molecule were patented and many studies came after, they found that actually the receptor responsible of this activity is actually a bitter taste receptor. So the TAS2 are one in the intestine. And so this looks to be the receptor responsible for this anti-diabetic property. So we have actually one drug candidate that target bitter taste receptor. So we are not it's not something that is not achievable. It's something that is already in place somehow.
That is great. I never thought that you would target taste receptors in the context of diabetes. It's very, very interesting.
Maybe we will know many more stories in the next future.
I am sure that we will. So you had also mentioned that one of the missing key pieces were tool compounds to study taste receptors and olfactory receptors. How would we get more tool compounds, or how would we? I guess you'd have to screen, but where do you start screening when you want to study these receptors?
Also, here there are many ways. My small contribution, the contribution from computational lab could be virtual screening. So you just screen because this, it's the cheap alternative to an high throughput screening. So you can have your models about bitter taste receptor model or just bitter taste predictor or odor predictor, and you can screen. I mean, now we have this huge library of molecules that can be used to prioritize or metabolites library and start. This is what we currently do, to start to find some hits and then to look around these molecules and look around this chemical space. But then there can be biggest investment, like really high throughput screening with this receptor, and that can provide indication from less focused area, just random. I mean, I think that in this stage, it's important where, I mean, the biological relevance of the molecule, but it's also important the chemistry to understand really how big is the chemistry that can respond to these receptors.
I haven't spoken about this, but I was wondering, what are the basic, 1 to 2 signaling pathways that are activated in, for example, taste receptors?
There is not a huge knowledge. I think that you are asking about beta arrestin signaling, or so what we know is that we have this gasteusin, this alpha protein that respond to this that signal with taste receptor. But for example, we cannot exclude that there will be something similar to other GPCRs. It's just that, I mean, the research stage is a bit in an earlier stage here.
The reason I was asking is because we were talking about, doing in silico screening and then moving on with some tool compounds or so compounds that give you hits on screen into a high throughput screening in the wet lab. And what do you look for when you do a high throughput screening in the wet lab? That's why I was wondering. I mean, you have to measure something, a physiological response in that sense. So it would be a regular, quote unquote, regular G protein activation or beta rest.
Yes, usually what it's measured is the calcium imaging using the regular G protein activation and the cascade.
Got it. Ooh, calcium imaging. It's a very tricky technique, definitely. All right. At the beginning of the conversation, you had mentioned that you started your lab at the end of 2018. What have been the challenges as a new faculty that you've been facing? Or what are the positive things of being faculty?
The positive things are easier, it's something that you are somehow, the academic career is always a lottery, you start and you never know where you end up till the day before you sign the contract. You never know if it will happen or if you have to change a different path. So of course the excitement to go on, it doesn't mean that you achieved already your your professional dream. I don't know how to say, because then you can always have different dreams personally, but I mean, it's already an accomplishment of something that was uncertain until the day before. So, you really say, okay, I'm here now, I made it. And this is already something very exciting to start. And then all the things that you were thinking in this perspective becomes true. So the recruitment of students and to be part of the institute management team, all these new activities that were not part of your routine becomes part of your agenda. And this is very nice because you start to have an active role also in the research management. And so this is very nice. And the challenge are more or less the same because you start doing something new that you don't know if you are prepared. I mean, no one is prepared, I guess. And so it's challenge to to be somehow in the other side. So just for example, in student recruitment, you were interviewed 1 month before, and 1 month after you are in the other position, and it's sometimes difficult to change this perspective and to start to understand things from a different perspective.
I don't know what it means to be a faculty, but the way I see it is it's an opportunity to contribute in a different way to a field, to contribute to the department that you're part of. At the same time, it is as stressful as setting up a new program or setting up a new experiment when you're a postdoc, and it's a high stakes job, and high stakes but high reward at the same time.
Yes, but in this I have to say that I was somehow also well prepared by my supervisor in the postdoc, because I have to say that Masha always trained me not only scientifically, but she also pushed me in this also with the collaborators. Somehow she always wanted me to be responsible of a decision, or also to participate in grant writing, not in an active part, but at least to know which is the procedure. And I was in a position that I could study her activities, and not just that everything. That I was in a ball, and then in a bubble, and then suddenly I went out of this bubble. So I learned step by step how. And I'm learning. I didn't get yet to a really professorship position. I still have small duties.
That's great. Yeah, and it gives you also the ability to see if you like this kind of career track. Having a supervisor and mentor that gives you other responsibilities than your own research, writing grants that you mentioned, but also taking part in decision making, allows you to, on the long term, see if you'd like to continue in this career.
Yes, and this is extremely important.
It is extremely important. So for scientists or young scientists, junior scientists who want to get into the field of studying GPCRs and get into academic positions, for example, what would be your advice to them?
I think to be passionate. Because it's really important to love this job, because otherwise it makes you very difficult to do it. To be persistent, because this is something that you learn with research. It's not something that you do something and after 1 month you get what you want. And if you are not passionate and you are not persistent, it's very stressful to do a job that doesn't lead results, but also to think of being an investor of yourself, So because I think that everyone should, a young scientist should read, should study, should do the most as you can to learn as much as you can, not only scientifically, but also networking, also the people who do your job. So to be passionate on exactly the field that you work on it, because this will be extremely useful later. And so to look at the future, to look at your future, even when you do your PhD, your postdoc, because the future will be later. So to be somehow visionary.
Definitely. A postdoc can end, the PhD ends, and you have to think ahead. And during these experiences, figure out what you like, what you're passionate about, and also figure out what you don't like.
Yes, this is what we were saying before. Not just doing things. You have your project and you have to do your project, but you have to think what is behind this project. Who are the people behind the literature of this project? What are the things that bring this project also finding, and to understand if this apply to you after, or if this doesn't work for you. And this is, because otherwise you just end up in a path that you didn't like.
Exactly. Agreed. What I wanted also to ask you is, throughout your career, you had mentioned that you came into close contact with GPCRs in Masha's lab. But throughout your entire career, were there any moments as a scientist when you discovered something or you learned something and you said, well, aha, this changes my perspective on the project I'm working on?
Yeah, I think that one is when I was think before to be persistent. This is what I learned during my PhD. I told you that I did this internship, if we can call, in Germany in a structural biology lab. And working with the computer, I mean, you don't get results, but you always have something, Working in the wet lab, it can be very frustrating in terms of results. So I was working in this project of crystallization, starting from the expression of the protein and so on. And the idea was to crystallize the carbonic anhydrase 2 with 5 molecules, small molecules. And I was there for 6 months. And what I got, it was only empty proteins until, I don't know, for 5 months, just empty proteins. We had an in-house X-ray, and my protein was always empty. And then 2 weeks before, I just changed it. I mean, I changed many parameters for 5 months. It's not that it's always the same experiment, but my successful experiment was 2 weeks before to leave. So it was almost the end. And then this was just an intermediate stage, because I mean when I saw my ligand, but I didn't have any more time to refine the structure. And then this was actually sent to the synchrotron. So actually my professor then allowed me to come back with the first postdoc with the research contract, just to finish the project. But for me it was, I mean, I think that was my happiest moment in my life, to see my ligand inside the protein and to see that I didn't give up because I was just finishing, because 2 weeks before you can just also pack your stuff and think, okay, I'm leaving. I didn't succeed. And it didn't change anything in my life. It's not that this really was a moment that I got some reward, but it was actually a life experience that really you should, if you want something, there is no time and no deadline, for it. You have just to try until the end.
You persevered, and look, I think it's a great personal accomplishment when you worked so hard on changing all these parameters, and it's not a trivial thing to crystallize proteins.
Yes, and of course for a computational chemist, this is the best thing, to have your protein, to really see what a protein means. It's not just download the file, but it's a lot of work behind, and to really understand what there is behind any coordinate. I think that was one of the most significant experience in my scientific background, competencies.
That's great. So perseverance, love what you do, and figure out if you love every aspect of what you do, and how much can you tolerate the things that are less pleasant but still get you to the goal where you want to get to.
It's also a nice exercise in general, because I don't think that there is something that you can love 100% of all aspects. You should feel comfortable and well to do it in any kind of decision you make, and this should be clear.
Definitely, yes, definitely, yes. There is that threshold of tolerance.
So it's important also to not underestimate, the job, or to have, this utopic imagine of your work or your life in general should be very. Because also this can create a lot of frustration after.
Yeah, I think everyone who has ever worked in a wet lab knows how frustrating it can be, and 99% of the time the experiments don't work, and then when it's 10 p.m. and it finally worked and you're the only one there and you want to share it, but still, thank God for social media. Now you can tweet or Facebook your gel or whatever you're trying to achieve. That's great. What I wanted to ask you also is if you have any job openings on your team, where can people find it? I just want to also mention that on the Dr. GPCR website, we have a career page. You're welcome to send us any job descriptions that you might have. It's free. We're just going to copy and paste them there and then put on a link to your email, for example, or to your website.
Yes, I will definitely do. We should have opening in the fall. Because my group is growing, but then this year we had this pandemic that somehow interrupted everyone plan, but hopefully we will restart soon everything. And usually I post everything also in my Facebook page, in my LinkedIn, Twitter, but I think that it's important to have this pages where you have more audience.
Definitely. And it's sometimes very hard to find, if you're looking for, for example, a non-academic position and you want to continue working on GPCRs, I find it very challenging to find these positions. So our goal with this career page is to aggregate all types of GPCR-related positions, whether they're postdoc, whether they're staff scientists, or whether they're industry positions, again, postdoc there, or any scientist or other positions that are related to the field so that people can go there and continue working on their favorite receptors.
And I was always looking for position, and I found very difficult, as you are saying, but it's also very difficult to find candidates. And this is something that I didn't expect, because just it's difficult to find the match. Because sometimes, so for example, I will start teaching just next year. So without teaching, I just rely on my colleagues, because there was no network for looking for students, for example. And this is, so it's so important to have some such initiative that try to match these two interests.
Exactly. And that brings me to another option that we offer. We also have a membership option. So it's also free. It's kind of your LinkedIn profile for GPCR scientists. It's drgpcr.com/membership. And the whole idea is that you fill out a form, you send us your picture, and then we'll post your membership so that anyone, for example, looking for students like you would be, without even beginning to teach yet, you could just go on there and look at members' profiles, and then it would be a match made in heaven if you're looking for somebody who has computational background, but also some GPCR background at the same time. I know we're in a COVID world these days, but usually, what kind of conferences do you like to go to? Where can people find you?
Actually, this COVID phase is not so bad for conferences because now many are going virtual. And this is also something so nice that I found, because, especially for young investigators, so for example, for my PhD student started recently. So, usually you go to the conference when you have something to present but now that everything is virtual, there is also a lot of possibilities to attend conferences as an attender. And this is something that I really think that is important, because conferences are not just made for the speakers, but also for people who want to understand this world, And so these are special occasions. So, there will be the Dr. GPCR Summit in September.
We're very excited about it, and we're excited to have it, and we're excited to be able to go virtual, to allow people to join in from anywhere in the world. And we're super excited. If people want to sign up, I don't know if you get a chance to register,
yes,
but it's drgpcr.com. Oh, great. We have already almost 200 people who registered, so from all around the world. So we're really excited. And yeah, hopefully we can continue on and create these annually.
Yes, and the other thing that is going virtual. So usually there is this European cost action that are made for thematic research network. So, and the one about signal transduction is called EARNEST. And also the next EARNEST meeting is also going virtual, and it will be in October 12-14. And this is also a nice event. Usually it's very informal. So I think that it is also tailor-made for young investigators. So there is a lot of space for early career scientists, and this I think that the next one is organized by early career investigators, but it's usually very high level. So this is also a nice virtual conference to attend.
We will definitely check it out. I think it's a good balance, having a high level conference with experienced speakers, but at the same time give the ability to young investigators, talented postdocs to present their work, and have it a little bit more democratized than your regular on-site conference where there is not enough time to give everybody the opportunity to talk about their fascinating research.
Yes, and this is the best environment also to build this network that is so important for everyone.
Yes, I agree. My best collaborations came out of discussions at poster sessions. Over a drink, and you get to talk, and you say, well, I know this person, let me introduce you, and you end up coming out with a cool paper and advancing the field. All right. Thank you so much, Antonella, for your time. It was a great discussion.
Thank you.
And
thank you, Yamina.
We'll talk to you soon. I want to also say to our audience and tell you as well, so people will be able to find this podcast's transcript and information, as well as links to your Facebook, your LinkedIn, and I think your Google Scholar, and any other ways to contact you. So if students, for example, who are listening, want to join your team, they'll know where to find you.
Many thanks.
All right. Thank you so much.
Thank you.
Thanks. Bye. Hi, Antonella. Welcome back. Thanks for being here today. I wanted to reach back out and talk to you a little bit about this whole COVID pandemic, and to see how you were doing and how your team was doing.
I mean, it was for everyone a surprise, So I'm molecular model. Basically, I work with my computer. I have remote connection, so everything can work perfectly wherever I am. But suddenly you realize that it's not so. I mean, that you have a social infrastructure that organizes your work, including the kindergarten, and then suddenly everything collapses. And this means that you have your computer, you have your remote connection, but you have also your 3-year son around, and also your husband working at home. So, it's really difficult to reorganize everything, and also to organize then a new lab. So a group that is under development. So my PhD students started in November, and in February we were in the lockdown. And this is, we managed very well. But it's not so nice to have an international student locked down in an apartment that is not house, is with no family, no friends, and work to do that is just the beginning of your work. I know that there are more serious situations, so we want to, but it was something that we were not prepared to.
Definitely. And how was the university's response to the whole COVID situation? I'm asking, for example, some universities, at least here in the US, from what I'm hearing, is that everyone was sent home to do remote work, and you needed to apply for a special permit if you were doing COVID-related research, or if you were doing something that was time dependent that would, make you lose years and years of data.
Mm-hmm. Yes, it was the same here. So our director organized everything. We are a small institute, so it was also not difficult to organize and prioritize project that was really urgent. So in our case, we are we have not this high priority to go back to the institute, because we can work from home with the computer. So for us, it was not a space problem. It was not physically. The problem was more the reorganization of the work in different times and different conditions.
Yeah, and also this is when you realize how much human connection is important, and how easy it is to peek your head into the office and say, hey, do you have a second?
Yes, and it's different to write a message and say, do you have a message? Because you're already bothering, with one message. And it's difficult to also have this group meeting virtually than physically. For something was also positive, because we said you need an organization, because you cannot just jump in the next door office. So, for example, with my PhD student, we had regular meetings that were very scheduled, and this, I felt that it was much more organized than doing just lively in the regular basis, because we had some daily feedback when we couldn't do daily, but it was very constant because we had to get organized, because we had to have a feedback each other. But all the rest, and it was just work, you lose all the nice part of your job. No coffee break.
No. Oh, virtual coffee. I think I was talking to someone last week, and they said that they were having virtual beer hours online. Not the same thing, but still very important to have those connections.
Yes, but I have to say that for me, I tried, but it was really difficult to manage everything, to include the social life in this already complicated family and work balance. Because I don't know, losing the kindergarten was like a shock. It's something that you don't, from one day to another, it just your workload increase of 100%.
Yes, especially with a 3-year-old and with a 5-year-old, or even with older children. I think the problem was that the schools closed.
The schooling.
And at least when they're young, it's not that big of a deal, although it's a lot of work. But when they have homework and they have to continue studying, and you have Zoom links flying all around every day, and if the school is not ready, it's also very complex.
And I also felt that the research work was more intense. I was involved in COVID-19 project, but behind this, I felt that everyone suddenly, we felt the pressure to work more, I don't know if this was the impression also, or also your impression, but I think that the intensity of work suddenly changed. And to have everything was really, it was one of the most intense working periods I have experienced, more than my PhD thesis. That is,
Yeah, I think it's a different rhythm. It's a matter of getting used to the new normal. And also, yes, you're always at home. So what else can you do other than working?
Yes.
And, it did happen to me that I woke up in the middle of the night saying, well, I'm thinking of something, I can't sleep. So, I don't have to, commute to work. I can just, get up at 2 a.m., work an hour, and then go back to sleep, which is not something you do before.
Now, you lose any schedule. Because Saturday is like Friday is like Wednesday, and I don't know, the morning is like the afternoon and the evening. So physiologically, in your days, you miss some reference point,
Yes, you do. And people still here, at least in the US, still tell me, well, have a good weekend. And I say, what does it mean? It doesn't matter. That's true. And I don't know about you, but I always have to look at the calendar to see what day and what date we are, because I feel like life stopped in March.
Yes, and it was, so my husband is teaching. So yes, online teaching, and yes, very random schedule. And each day I was asking him, I have his schedule in my calendar to remind, because we had to, of course, to adjust our time. And each day, each hour, I was asking, where are you teaching? When are you teaching? And I say, you are a scientist and you cannot remind some, but it was really impossible for me to remember times and days, and I had always to look at it, because it was, every day was the same. So I was completely lost in my work, environment. And I felt also bad, like a mother, because also having your son together with work, you completely lost the quality time. You just give food and then television and then sleep and then do something, but you never have, I don't know, this time for walking, or, I mean, we had it sometimes, but you never feel, I mean, that now the work time is over and now it starts the family time, because everything was together.
Yeah, I agree. That's the hard part. I think what kept me a little bit more on time was we have a dog. So walking the dog is essential, and he knows what time we're going. So he knows feeding time, and he also knows walking time. So that gives you an idea. But then again, it doesn't matter what day you are; you still have to walk the dog and feed the dog. So it was a little bit of help.
These are things that maybe we never stopped to think of, but are really important thing in, I mean, in the flow of your day and of your week, to have such, I mean, standpoint in the day.
For the first time, I think for a lot of us, not the outside environment, but our own clock made us determine what we need to do and when we need to do it. It's definitely a brand new world in that sense. Did you do anything special with your team to keep morale high, or did you do like Zoom meetings that were not about science? I know you had mentioned that it was difficult, but you still had more meetings. Anything you can tell us?
Yes, I mean, our meetings are never so formal. So yes, we always combined. I mean, for now I have just one PhD student, so it's very easy to keep to have work, but together to have also, some other of discussion. And so we shared feeling and problems. I mean, I think that it's a very open relationship. So as I mentioned, it was also challenging for him. So I gave him some time to get organized with this new schedule, because I think that it's frustrating to have the family at home, but it's also frustrating to be suddenly alone, while you were building your new social life. And this is also difficult to suddenly, be at home and having only your work. This is also not something healthy as well. So we tried there was nothing formal to discuss all these things, just randomly to check how we are and what we do.
Yeah, of course. And so how are things now? Are you planning to go back to the office? Are you reopening, or you're still working from home?
So the institute reopened in the middle of June.
Okay.
But together with the lockdown, we have also so a relocation of the building. So our office were ready just this week, but the policy is to have still part-time. I mean, we will work 3 days per week in the institute, and we can work at home the other days. But as computational people, we are not, for us it's not so important really to be back in the institute to work, but we are going back to work from next week, as you mentioned, to have a social network, to go back, because it's different to say, okay, we are here, this is our working place, and to reorganize the things also in a different space and not just at home. It's really different to work at home. I always like it to have this home office, home working, because when you have a lot of work at home, you can really, just focus on work. But on a long range, focusing only at work, it's really dangerous. Even for the work itself, is not only because, you need to have a breathing, and to have fresh air also for your brain to think of what you are doing, and not just to do it.
Yes, I agree. I think you get your best ideas in the shower, not necessarily sitting in front of the computer. All right, thank you so much, Antonella, for taking the time. We wish you all the best. Hopefully,
and hopefully, yes, we will go all back to work regularly soon.
Yes. And I wish you good luck going back to work next week, and we'll be in touch.
Thank you.
Thank you. Bye.
Thank you, Amina. Bye.
Thank you for listening to this Dr. GPCR podcast episode. We hope you enjoyed it. If you'd like to sponsor us, please visit drgpcr.com/sponsor. Your support will allow us to bring you more podcasts and exciting GPCR content. Next time on the Dr. GPCR podcast, our guest will be Dr. Amina Pradhan from the University of Illinois at Chicago. Thank you to Dr. Antonella Di Pizio and Attila Forrest. Music by Rosa Bershish. I want to also say thank you to Jin Chong and Shivani Sajdev, who are our talented, dedicated science communicators. I'm your host, Dr. Yamina Bershish. Thank you for the privilege of your time, and until next time, stay safe.